structural biology and biophysical techniques allow to visualize the molecules of life and characterize their properties. Application of such techniques is the role of the Center of Molecular Structure, which is a part of the Institute of Biotechnology of the Academy of Sciences of the Czech Republic. X-ray methods reveal the three-dimensional structure of proteins, nucleic acids, and their complexes using X-ray diffraction and scattering. Single crystal X-ray diffraction allows visualization of molecules at atomic level. The crystallization robot is used for growing crystals with dimensions of tens to a few hundreds of micrometers. X-ray diffraction is measured by directing a beam of X-rays at a crystal. This produces diffraction patterns recorded on a detector. The enlarged image of the molecules in the crystal is computed using Fourier transform of the diffraction pattern, which helps to understand chemical basis of interactions. This information is used, for example, in the design of novel drugs or in biotechnology. Small-angle X-ray scattering enables visualization of protein structure at low resolution without the need of a crystalline sample. Other techniques, such as circular dichroism, dynamic light scattering, or calorimetry, provide important data about structure, structural integrity, and stability of proteins. Information about interactions of proteins with other ligands or with each other can be revealed by surface plasmon resonance or thermophoresis. Structural mass spectrometry allows to map protein structural changes, protein-protein interactions, or protein small molecules interactions. Extreme resolution and precision analysis of biomolecules is done on a very powerful tool, the state-of-the-art high magnetic field Fourier transform ion cyclotron resonance mass spectrometer. The Center of Molecular Structure is a part of the BioSev project and provides services as a member of the Czech Infrastructure for Integrative Structural Biology and of the European Infrastructure Instruct.